It's recording.
Yes. Blue right now has 70. White has 50. Pink has 40. Orange has 40. Purple has 40. Green has 50. White has 50.
How much do we have?
All right, we're talking about enzymes. Tomorrow we're going to start an enzyme lab.
Yeah. Are we going to finish on this?
Probably not. Start today, finish tomorrow. That's an enzyme. You know what it's called?
Enzyme.
Catalase. Can you say catalase? Catalase is an enzyme. Can you tell that's a protein first by looking at it?
Yeah.
You see it's all folded up. You see any alpha helices?
It looks like one of those.
See all the helices? You see the the pleated sheets? These blue areas are pleated sheets. It's a big molecule. It's all folded up and takes on a certain shape. They call it catalase. And what catalase does is it breaks down hydrogen peroxide. You have to learn a little bit about chemical formulas now. You ever heard of a chemical formula? Pay attention over here. Ladies up here. Hydrogen peroxide, you may have heard of this, is a chemical that has to be broken down if it builds up in your cell. Hydrogen peroxide is poisonous to your cell. And your cells have to deal with that. And so there's a special enzyme that will break down hydrogen peroxide into water and oxygen. And that is not a balanced chemical equation. To make it balanced, you say that two hydrogen peroxides form two water molecules and an oxygen. The things that you start with in a chemical equation are called the reactants. And what you finish with in a chemical equation, you know what those are called? Have you learned this before? The products. Have you ever heard the product of a reaction? That's what you end up with. Did y'all ever do a chemical reaction in middle school?
Yeast.
You did yeast?
Is yeast alive?
I remember balancing.
Is yeast alive? You did balancing equations? What class would that have been?
Physical science.
Tiganis? Okay. Good. So y'all have seen something that looks like this before. Okay. So you start with the reactants, you end up with the products. Let me show you this with my models. Let's start with two H2O2 molecules. I can build those pretty easily here. This thing right here. Let me take off the electrons so we can see it a little better. Can you tell that's H2O2? Those are H's, those are O's. H2O2, that's this. And you see how I have a 2 in front? That means I have two of them. So let me get another one in here. Are y'all watching this? This is fantastic. Do you see how that molecule... no. Okay. Do you see how that molecule is H2O2? It's got two H's and two O's. That is an H2O2 molecule. Everybody see? The 2 in the front means I got two of them. An H2O2 and another H2O2. We're going to do a chemical reaction here. This line means a reaction happens that changes these into two H2Os and an O2. You want to see that reaction happen?
Yes.
Break these apart. Put these there. Hook those together. Now what do we have? Two H2Os. Can you see those two H2Os? And, that plus means and, an O2. That was a chemical reaction.
Are the O's double bonded?
The O's are double bonded to one another.
That's pretty cool.
You want to see it again?
Yeah.
Yes.
Woo! Is this your favorite thing?
Yeah.
See, I told you. I didn't tell you, but anyway. Here we go. Let's try it again. Two H2O2s. There's an H2O2. There's an H2O2. React. La 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 la.
I told you.
Blah 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 blah. Boop. To form two H2Os and an O2. Wasn't that a cool reaction?
Yes.
Now, up here on the board, I'm moving it around with my fingers. But in your cells, there aren't fingers moving around, tearing up molecules and putting them back together. Did you know that? There aren't fingers.
What?
There aren't people crawling around in your cells, grabbing things and moving them around. You know what there are?
Enzymes.
Enzymes. There are proteins. Instead of people, there are proteins that do this. There are proteins that grab the H2O2 and tear it apart and put the other things back together to make these products. There are enzymes that do that. And the enzyme that does this is called Catalase. That enzyme carries out that reaction.
How do you spell Catalase?
Great question.
Catalase.
And usually they'll write it above the arrow. Catalase. Catalase is the enzyme that carries out the reaction. There's another one. The one we're going to use, there's another enzyme that does it too. It's called peroxidase. Peroxidase also carries out that reaction. That enzyme they're showing there is called catalase, but peroxidase is a different enzyme that can do the same reaction. Tomorrow we're going to work with peroxidase.
That's a chemical reaction.
So bring your lab notebooks tomorrow. Yes, this is a chemical reaction.
Okay.
The tearing apart what I did up there was a chemical reaction.
Okay.
And what do we call the stuff that we start with?
Reactant.
And what do we call the stuff that we finish with?
Products.
All right, we good so far? And why do we want that reaction to happen in our cells? Why not just leave it H2O2 and not break it down? It's poison. It'll kill the cell. So we got to break it apart. Your body cells are full of enzymes that break things apart and put things together. That's how you function. That's life. Biology is the study of life. So how does a cell maintain itself? One thing it has to do, one of the many thousands of things a cell has to do, is break down hydrogen peroxide whenever there's too much of it. That's one of the things your cells do. There's thousands of other things they do. Well, you just learned about one of them. Wasn't that great?
Mm-hmm.
All right. Moving on. You need to understand this graph. This graph shows the progress of the reaction. Here's the start of the reaction. See, it says reactants. This says glucose and oxygen, but you can substitute H2O2 for those. And here the products are on the right. So over time, the reactants change into the products. And the big humps that you see here, that's the energy that's necessary to get a chemical reaction to occur. We call that the activation energy. I need a different color marker, y'all. Where are my other colors? Hold on. Let me go over here. Here's some other colored markers. Back when I was a kid, they didn't have color markers. They had chalk. We didn't learn anything. It was terrible.
Does it have colored chalk?
Okay, so here we go. So, why did I get another colored marker? Oh, activation energy. Activation energy is necessary to get this to change into that. You have to give it some energy. This thing will not change into those things unless it's first given some energy, and we call that activation energy. Now, if we were going to change this into those, we would have to give it a whole lot of activation energy if we didn't have an enzyme. So this says without an enzyme. Without an enzyme, you have to give it a whole lot of energy. And usually the way this is done in a lab is you heat it. If I were to take hydrogen peroxide and get it real hot, put it in a beaker and put a flame under it, the hydrogen peroxide would get so hot that it would come apart into those things. This molecule would just start vibrating really fast and then would break apart and end up these two things. But that would take a whole lot of activation energy. You see that big hump? That big hump represents a lot of energy that's needed to go to the molecules. But with an enzyme like catalase or peroxidase, you don't need nearly as much energy. You can break down hydrogen peroxide at a much lower energy. And you only have to overcome a little bit.
So you still heat it?
It'll still heat it with the enzyme, but not nearly as much heat.
So you still need heat?
You still need some heat. And you notice that your body's warm? Your body heat supplies the heat for your chemical reactions to occur. You're breaking down food and gives off some heat, and that's enough. So a warm-blooded creature that keeps its body temperature high can do a lot more reactions than a cold-blooded creature, like a lizard or a snake or something like that. They have to actually heat their body up before they can carry out chemical reactions. So they go lay out in the sun or something like that. You noticed that before? Yes. Have y'all noticed there's all kinds of lizards running around outside? Whenever you walk on the sidewalk?
They're really little.
Yeah, they're everywhere.
They're like
Right
skeet.
when you're walking.
Yeah, I don't know. Yeah, they're like blended in, and then you walk and it scares them and they move. But that's pretty cool. Maybe we'll do. Maybe we'll study their respiration when we get to that section.
Lizard hunt.
Yeah, we can pull some lizards up and take their see how fast they're breathing. Then we can heat them up or cool them down and see if that changes their rate of respiration. Not to kill them, just a little bit of heat. Give them a little bit of warmth or a little bit of cold, and see if that changes their respiration. Wouldn't that be a good experiment?
That would.
Let's do that. Okay, so let's watch this little video of activation energy, and then I'm going to ask you a quiz question, or a priority question on it.
Making a snowman is always a big ball of fun. 3 snowballs full of fun to be exact. And each snowball starts out small, getting bigger as you roll.
Let me start over.
Making a snowman is always a big ball of fun. 3 snowballs full of fun to be exact. And each snowball starts out small, getting bigger as you roll it in white fluffiness. Let's imagine that you go outside in a fresh snowfall to build one of these guys. But you don't have the best suited yard for rolling snowballs. In fact, your backyard is one giant hill. This hill is fantastic for sledding, but not so much for making snowmen. Anyways, in order to make a giant snowball for the snowman's bottom, you'll need to actually push the snowball up over the hill and then down the other side. You do it because it's fun, but make no mistake, it takes a lot of energy to get started up this hill, and this is hard work. Right now, you share a quintessential problem with some chemical reactions within a cell. These chemical reactions might be a lot of fun when you're in a cell, but it takes a lot of energy to get them started. This is called the activation energy, or the energy required for a reaction to start. You can think of activation energy as that energy you needed to muster up in order to push that snowball up and over the hill in your backyard, in order to maximize the size of your snowball and get it to the other side. What your cells have to help overcome a problem of high activation energy are called enzymes. Enzymes are proteins that lower the activation energy of a reaction. In doing this, enzymes increase the rate of a reaction, helping it to occur faster. However, enzymes are not consumed in the reaction, they simply help it to occur. They do not add snow to your snowball, they just make it easier to make a snowball. This makes enzymes reaction catalysts. The presence of enzymes is analogous to decreasing the size of your hill in your backyard. Enzymes make things easier for your cell and help chemical reactions occur. There are hundreds of different kinds of enzymes in your cells, which all participate in different types of reactions. Enzymes can break molecules apart, build or add molecules, and even rearrange them. In lowering the activation energy of a reaction, enzymes decrease the barrier to starting a reaction. It's important to note, however, that the change in energy remains the same between the start and end of a chemical reaction. For example, if a reaction released 200 kilojoules of energy without an enzyme, the same reaction would still release 200 kilojoules of energy with some enzymatic aid. The difference would only be a lower activation energy and a faster rate of reaction. In other words, if enzymes were helping you build a snowman, you'd be done quicker, but you'd have just as much fun.
All right. Y'all understand that? So 10 points. 10 points if you can tell me, as far as activation energy goes, what is the purpose of an enzyme? What is the purpose of an enzyme in a cell? 10 points. Write it on a sheet. What's the purpose of an enzyme in a cell? Talk about it in terms of activation energy.
You're left-handed?
Okay, purple correct.
It uses the same amount of energy. So what's wrong? Okay. Oh, Erica, sorry, you need to trip you.
How's it going here?
Great, do you want to film it? I don't want to anymore.
Okay, I'll give it the pink.
Do you want to do it? Yeah. Hey.
Yes. Orange is correct.
Yes. Do you want to do it?
No. White. Yes.
Green.
Yes, blue is right. Green, anything?
Yes.
Green.
I watched that whole thing unfold.
It's not easy being green. Remember that?
Wait, Adam, did we get it right?
Muppets? Green is correct. Yeah, Kermit sang a song called "It's Not Easy Being Green." It lowers the activation energy. And enzymes lower the activation energy, which makes
Do you want to record?
reactions occur faster. Question?
Don't worry, I won't get you in it if you don't want to be.
Yes. You don't want to ask? Rhea, you can ask whenever you want.
Lucy, you want to record?
Don't make fun.
Yeah, very enjoyed. This was a bullying video.
Okay.
My class didn't watch the bullying video.
All right. So let's see how an enzyme works here. Enzymes have an area on them called the active site. Can you say active site?
Active site.
And the thing that fits into the enzyme is called the substrate. Can you say substrate?
Substrate.
So in the case of our reaction here, the enzyme Catalase has an active site. Now remember, an enzyme is a protein that's all folded up to make a certain shape. So that's what this thing represents, a folded protein, a protein that's folded into this shape. And the substrate there fits into the shape just right. So that substrate might be the H2O2, the reactant. The substrate is the reactants. That's the thing that fits into the enzyme and fits in such a way, and then the enzyme will break it apart and release it. And there are the products. Notice that the enzyme isn't changed in the reaction. The enzyme stays the same as it was in the beginning. So the enzyme just helps the reaction happen because it has the right shaped active site to allow the reaction to occur. In other words, as soon as this thing fits in there, it maybe see how the enzyme has a little wedge right there? See how that little triangle? If this thing lands on there, that little triangle there might push against that bond there and break them apart. It's like the enzyme has just the right shape to break that up. Y'all see what I'm saying? It's all about shapes with enzymes. This is a big protein, and there's the substrate. The substrate is what comes in to make the reaction occur. Let me show you some video footage of this. The videos show it real well how enzymes work. And see if you can follow what's happening here. I might ask you some questions. I will ask you some questions about it. So, it says enable all not recommended. I'm going to do it anyway.
The enzyme attracts the reacting molecules or substrates to its active site, then releases the end products. Its effect is astounding. Each enzyme can assemble hundreds of thousands of new molecules every second.
How cool is that?
Looks like a cactus.
They just like hit it and then it changes.
Yeah, so here we go. So this big green thing, did they show this from a distance? Yeah, here we go. So this big green thing down there, that's your enzyme. That's your protein. It's actually a protein made of a bunch of amino acids. That's this big green thing. And it has just the right shape. So we call this area where the reaction takes place, it's called the active site. Can you say active site? Come on, y'all. Active site? Wait. Can you say active site?
Active site.
Much better. I gotta get more fired up about this. Okay, so here we go.
The enzyme attracts the
So I can do the talking. There's the substrates, and then they react to one another and form something new. Two things come in, they react, and go out. This area where they're reacting is called the active site of the enzyme. Notice the enzyme doesn't change. So peroxidase is sitting there doing this. Hydrogen peroxide is coming in, and water and oxygen is going out. The peroxidase can do this so fast. Listen to this number. The peroxidase can carry out 300,000 reactions per second.
Whoa. That's
Per second.
crazy.
In 1 second, it can do it 300,000 times. Isn't that incredible?
Wow.
Yes, because on a small scale.
It's very tiny, but small molecules move very fast. Think of this in 1 second doing this 300,000 times. That's one. That's two. That's three. Think 300,000 times in 1 second. Four. That'd be a lot of reactions, right?
Yes, sir.
Amazing. How about this one?
Substrate molecules bind the active site of an enzyme molecule.
This one's slow.
After the enzyme-substrate complex has formed, catalysis can occur, followed by the release of the product or products of the reaction.
Okay, there's a new word for you. So, when the enzyme is bounded to the substrate, we call it the enzyme-substrate complex. That's the enzyme together with the substrate. Enzyme-substrate complex.
Catalysis can occur, followed by the release of the product or products of the reaction.
Do you see how that's different than what we started with?
Yes, sir.
The green thing is different from the green and the blue thing. The green and the blue thing come in, and just one big green thing goes out.
Do we have to know what catalysis is?
Catalysis is just a general term for a reaction.
Is it a catalyst that speeds up a reaction?
That's right. Uh-huh.
An example of how enzymes function in the body is from the enzymes.
I'll do the talking, sir. So, do you recognize this system here? That's your digestive system. How many ate something that had sugar in it for lunch?
I
did.
Yeah, I did.
Everybody did. So this is happening right now in your body. Let's see how enzymes work inside your digestive system. So if you go down into your intestine, this is your intestines. If you look in your intestines,
it's a carbohydrates.
here's the inside. Here's what the inside of your intestine looks like. Are y'all with me here?
Yep.
Inside of your intestine.
I may be wrong though.
It's got all these kinds of folds and things. And then these little finger-like projections here, they're called villi. And then on the surface of those villi are little hairs called microvilli on the surface. This is deep in your intestines now. And on the surface of these microvilli, you have little enzymes, little proteins. And they're showing you one of them up close here. And this enzyme here is called sucrase. And you know what it breaks down?
Sucrose. Base.
It breaks down sucrose. This is sugar. Watch how the sugar will fit into the enzyme here, along with the water molecule, and bam, be broken apart. And now we have the products, glucose and fructose. So this is happening right now in your digestive system while we're talking. Sucrose is being broken down into glucose and fructose. And it'll just happen over and over. And this is how you digest your food. You've always heard about digesting your food, but I bet you didn't know how it happens. This is how it happens. You got enzymes in your digestive system that'll break it down.
Wait, what is that little tail?
Does it
That
actually
little
look like
tail
that in your stomach?
bends over when everything binds. The little tail bends over and helps the reaction occur.
So sucrase breaks stuff down into sugar?
Into glucose and fructose. It breaks sugar down, breaks sucrose down into glucose and fructose. Remember the disaccharide? Here's sucrose is a disaccharide, and it breaks it down into 2 monosaccharides with the help of water there. Now we have 2 monosaccharides leaving. Make sense?
Yes.
All right, I got questions for you. You ready? For you and your partner? Okay, so here we go. This is sucrose. It is what starts the reaction. So what do we call sucrose? In this case, what is another word for what starts the reaction? 10 points. It's what you have at the beginning of the reaction. What's the word for that? What we call sucrose here. Sucrose, remember, is going to go into the enzyme. Blue is correct.
What do you say?
Anyone else? What would you call sucrose here? Yes. Orange is correct. Green is correct. White is correct. Anyone else? Yeah, close your computer. See if you can answer this without using your computers or notes. See how much you learned. So we call sucrose the reactant. It's what starts the reaction. It's what we start with. Now the reactant sucrose here will fit into the enzyme. What is the word for something that fits into an enzyme? 10 points if you can tell me there's a science word for something that fits into an enzyme. Scientific terminology, baby. Scientific terminology.
Yeah, that's what it was. I couldn't think of the name.
Yes, blue is correct.
Oh.
Orange is correct. Thank you. You can just set it face down. White is correct.
Hold up.
Green is correct. Anyone else? Hey, y'all don't pack up yet. We got plenty of time. Purple correct. Pink correct. It's called the substrate.
Oh, and you're not even. Oh.
It's the substrate. Now I'm continuing on. Watch this. The sucrose is attached to the enzyme. What do we call the whole thing right now? 10 points if you can tell me. What do we call the whole thing right now? Write it down. Write it down. What do we call the whole thing when the sucrose is connected to the enzyme?
Boy, let me see that.
We have a name for that whole thing.
Okay, yes. Adam, be secretive when you do it because there's some snakes.
Yes, orange is
you guys,
correct.
but who knows if there's snakes?
I'm a part of your
Anyone else?
I know, I didn't say you.
Yes, green is
I
correct.
never knew my group was said. I'm the lemon.
You are a.
White is not correct. Purple is correct.
Girl,
I don't even watch these.
Blue is correct.
Now your hair looks like.
I don't watch
Okay, hang with me here. Oh, pink.
What the heck?
Just mix these two.
Pink is correct.
It looks like a dog.
It's a saucy salad. It looks like anything.
Okay.
I thought your finger.
Y'all pay
You know, it's like
attention.
little
Mary Jo out
players
there.
that have like a dog on it.
This
or
is
something.
called the enzyme
what they're called.
substrate
I'll send you a
complex.
picture of these in style. They're like the
Now, 10
players
points
that
if
were
you
like
can tell
dogs.
me, what is this area called where the substrate will stick? What is this area called right here? 10 points if you can tell me that. There's an area where it attaches.
I'm not videoing you. I'm trying to close this.
Yeah,
What's the
blue is
question?
correct.
It's an active salad.
Anyone else?
Yeah, you can be in our group, Maria.
Green is correct. Anyone else?
I need some freaking eye drops. My eyes
Pink is correct.
itch.
Going.
Wait, you're writing it. Yes, Adam.
Orange is right.
Ooh. Hey, Adam, let's do a fist bump then high five. Ready? Ooh. Ooh.
Aw. No, it's ew.
How about high
I
five?
can't give it a purple, but I won't count it wrong. It's called, listen, it's called the active site. Active site.
Thanks, Maria.
Okay? All right, we're moving forward. You need to understand this graph. Listen, yeah, you can open your notes back up. You need to understand this graph. Every enzyme has an optimal pH that it works at. Y'all remember what pH means?
Yes.
What does it mean?
Per hydrogen.
Yeah, what does that mean?
How much like acid or base is
How
inside?
acid or base it is. Very nice. How acidic
I
or
think I
basic
deserve
it
10
is.
points for that.
So,
I'm kidding.
every enzyme has an optimal pH where it works. The enzyme that they're showing here has an optimal pH of 7. That means this enzyme has the best activity if it's pH 7. If the pH gets too low, it doesn't work well. See, this is the rate of the reaction, how fast the reaction occurs. If the pH is 2 here, that reaction won't happen at all. If the pH is 12, the reaction won't happen at all. If the pH is 10, the reaction will happen, but not very quickly. If the pH is 7, that's just right.
So why is
It's like the Goldilocks.
Why is the pH of blood 7.4?
Because it's got some baking soda, sodium bicarbonate in it, which is a low pH, and it acts a high pH, and it acts as a buffer to prevent the blood from getting too acidic.
So that accounts for the 0.4.
Found it.
So the pH of some enzymes work best at pH 2. Like, for instance, all the enzymes that are in your stomach that break down the food when it's in your stomach, those work best at acidic pHs. And so if we were to graph those, there'd be a big hump here at pH 2. And some work better at basic pHs, like the ones in your intestines work best around pH 8 because your intestines are a higher pH. So it depends upon where you are in the body as to what enzymes work the best. But it has to do with enzyme structure. The enzymes are pulled upon by charged particles like hydrogen ions. Do you remember if something has a low pH, it has a lot of hydrogen ions floating around? I'm losing some of y'all. Y'all hang with me. There's only a few minutes left here. If something has a high pH, it has a lot of hydrogen ions. I'm sorry, a low pH. It has a lot of hydrogen ions. If something's very acidic, it's got a lot of hydrogen. The hydrogen ions, because they're charged, will pull on the structure of the protein and can pull apart the protein and denature it. And so that is why at very low pHs, an enzyme might not work well, because the hydrogen ions pull on it and break up the structure. And at very high pHs, the hydroxide, the OH minus ions, can break up the structure. So you want, most enzymes work best at some certain pH where they're not pulled on too much either way. Interesting. There's also an optimal temperature for every enzyme. If the temperature is too cold, you won't have many reactions occurring, because molecules don't move very fast when it's cold. So, have I told you before that molecules are always vibrating?
Yes, sir.
And at cold temperatures, they vibrate slowly. And guess what they do at hot temperatures? They go real fast. So, at cold temperatures, we have a low rate of reaction. As you heat it up, the molecules vibrate more, and so you get faster reactions. But when you'll reach an optimal temperature. But if you get hotter than that, the protein itself starts vibrating too fast and breaks apart. And so you get less reaction there. And so there's some optimal temperature that it's not so cold that the reactions aren't happening much, and it's not so hot that the protein comes apart. There is a perfect Goldilocks temperature. Y'all know the story of Goldilocks? Do they still tell that?
I know it.
Kids? Yeah. This porridge is too hot. This porridge is too cold. This one's just right. It's one of my favorite bits.
Is there a different temperature for every enzyme?
And different optimal temperature, yes. Each enzyme has its own optimal temperature. Some enzymes are tougher than others, and it takes more heat to break them apart. And some enzymes don't have very strong bonds, and just a little heat will tear them apart. So, yeah. All right? Are y'all good?
Yes.
Have you ever looked at the side of a cereal box and seen all those things in it? Vitamin A and C and iron and vitamin D and zinc. Why do you need all that stuff?
Zinc prevents colds.
Well, why? You're right, it does. All of these things are used to help make enzymes. There is an enzyme that requires zinc in its structure. The enzyme doesn't make the right shape unless you have some zinc in the structure, and then the zinc makes it have the right shape. There's another enzyme that requires vitamin A. There's another enzyme that requires magnesium. And so all these things that you need in your diet are to help build your enzymes. You don't need much of them, but you need some of them. What were you going to say?
Oh, is that like a cereal box?
Yeah, this is like the side of a cereal box or something.
A mountain cereal, it says like on the bottom.
A mountain cereal, yeah. I don't know what cereal this is.
Lucky Charms.
Is it?
We can say it is.
Always after me, Lucky Charms.
I love Lucky Charms.
I do too.
I don't. I like the Charms, but I don't like Lucky.
See, so all this time you were alive, you know that you need vitamins, but you don't know why. Now if somebody asks you, why do you need vitamins? Don't just say, to live. Say, the vitamins help build the structure of the enzymes I need to carry out my chemical reactions. Isn't that a better answer?
Yeah. Gracie, you know some cereals are better because you put like
All right. Thank you. You can turn off the video now. And